centers that we have in the country have played a very important role. So I would like to request Dr. Taslim Arif Saeed, CEO of CCAMP. Taslim? Yeah, there you are. To please lead the discussion. We have with us as panelists Dr. Aseem Mishra. Aseem was also an early career fellow of India Alliance, and he's now the CEO and co-founder at Prante Solutions in Orissa. Dr. Dipanita Chattopadhyay. She's the chairman and CEO of IKP Knowledge Park, Hyderabad. And we also have with us Dr. Vijay Chandru, founder director, Strand Life Sciences. Thank you all. Thank you to the panelists for coming today, and over to you, Taslim.
Good morning, everyone. Good morning, everyone. It's a pleasure to be here. And I've been not able to be here, but I have been following the talks through the social media and other aspects. I think that's the great boost for what happened. But thanks, organizers, Shahid, Sarah, and others to consider this session, which I feel very strongly about, largely. And I have some reasons that make me feel strongly. As you know, this session is about the translating your discoveries, and I have taken, I don't know whether we are being showcased, I've taken liberty to use 2-3 slides just to set the context, but I also taken liberty, Shahid, to add one word in the given panel title. It's translating your discoveries in and around incubation centers in and around academic institutions. So, if we have fantastic panel here. We have, if I may start from right, we have Vijay Chandru, who has been an academic turned entrepreneur, phenomenal role model, and what he has done. I think the introductions, I'll have them introduce themselves. Aseem, who has been a fellow himself before, and he has been connected with an incubation center and now full-fledged co-founder, if I may say so. And of course, Dipanita, who has been spearheading the entire phenomena of bioincubation per se, so it is through IKP and so on. But if I may say so, in terms of, and I just heard Sarah say that there has been discussions over a few days over translation. When we look at it, of course, people talk about that how much percentage of our GDP actually goes in science, right? And people talk about this country does 3%, this country does 2%, and we do possibly around 0.75 or 8.8%, if I'm not wrong, of GDP. G2 is suggesting even lower, and it's going down. So while we do that, if we do science per se, and even take out the DAE and other funding, even defense funding, the numbers that come around, and there is no collected number, but the numbers that come around comes around 15,000 crores odd. It could be 15 to 20,000 crores per year if you take the funding. Now, what happens, and this is where I'm coming, what happens with this phenomenal funding? There are two great outcomes that actually come out of this 15,000, 20,000 annually that goes in. One, very, very important, phenomenal scientific training that happens, the human resource that we generate. Very, very exciting thing. I think we train very good scientists at different levels and help them build their career at the next level. And that cannot be, it's not trivial, that is phenomenal, and I think that gives an edge to us. The second very exciting thing happened is that very, very good science and very, very good knowledge that comes out of it. If I use the yardstick that NASA actually attempts to look at it from the idea to market, because in terms of science, so NASA uses something called technology readiness level, and they say, okay, let's do the journey and give them the 10 steps to it. So it's 0 to 10, and when they look at it, it starts from absolutely an idea generation over possibly a coffee session or in your while you're having a shower or you're in discussions, to all the way when it is launched in the market for public consumption, if I may say so, and the use and impact. This is 1 to 10. What happens with the funding here, it is my opinion, and it has to be corrected if I'm wrong, we actually produce very, very good science in the technology readiness levels 0 to 3. That is the early, that starts from idea to proof of concept. It's very, very good. Phenomenal ideas are generated, they have proof of concept, and what we generate, if I may say so, is we generate promise. We generate very, very good promise, which has a potential. And that promise and potential actually has to be taken forward. What we would call it as a translation, to actually achieve that promise or a potential. And that is a very important, crucial journey that has to be taken. And I think that is something very, very important. If I may request to use the slides if they're on, I have just intended to depict the same in 2 to 3 slides, not more, but this is a broad standard roadmap. This is nothing new, but I just want to set the context. We start with a scientific idea. We have deep science discoveries that come out of it, which leads to, upon translation work, deep science innovations, which then go on to this commercial products. I don't think in this room I need to say that there is no point to actually devalue the commercialization, because upon commercialization, it reaches actually when it's supposed to reach, that is the public and the societal impact. The next point, if I just say that what is happening is it is a global phenomena, a funnel effect, when you want to cultivate the science-based innovation, where you have very large numbers. I think I would say that India, we need more and more, and hence what Jitu indicated, we need more funds to have more basic knowledge so that more gets translated and have more industrial applications. The last slide, and nothing more, but this is what actually happens. This, if you actually convert the same last 2 slides, which has the same content, it actually, there is a TRL level, which I'm sorry about the colors that are coming, but if you see what academia generates is a great science, excellent science. The content requires the same thing, but when you talk to industry, what industry wants is the same science in a different form factor. That is a validated, de-risked, further advanced form of the same science. That is phenomenal understanding, the basic knowledge upon which we can build it. And that is something I think we should work on, and saying what models can actually use to intervene at that stage and go forward. At CCAM, we are attempting to build a model that we have had a pilot. I'll not go into it for now, but we feel very strongly about it. And today's panel allows us to actually have other views in terms of what we attempt to do. So with that context, I think we can let these slides go. I will request my co-panelists to actually introduce themselves briefly. Again, the panel has been set to build it, but what we attempt to do is that upon introductions, we possibly talk about the models, the way the translation is being done in and around in academic institutions, and what possibly we can do in that model. So if I may request Chandru to actually take on it. Yeah, let's go.
Thank you, Taslim, and very happy to be here to address all the fellows and other attendees. So as probably many of you know, Strand Life Sciences was a translational enterprise that came out of the Indian Institute of Science. It's actually the first example in the country of faculty actually, stepping out and starting an enterprise. So we were the guinea pigs. We were 4 professors in computer science. Not many people know this, but the Indian Institute of Science was the hotbed of machine learning back in the 90s. Lots of the early developments in machine learning actually came from the Indian Institute of Science, particularly in the Indian context. So we saw an opportunity to come into the life sciences with that expertise in machine learning, and we thought that the 4th paradigm of science was actually starting to affect the life sciences, particularly with projects like the Human Genome Project. And so we thought it was time to step out and translate in the life sciences. And of course, as it turned out, I think the industry in biotechnology, in pharma and biopharma, was not quite ready yet to absorb the kinds of tools that we were building. But researchers were. So for the first 10, 12 years, all the products and the services that we built was directed towards the research community. And, you know, some of the tools that we built actually have resulted in about 25,000 citations. So in publications, so tools that you might be familiar with like Gene Spring and Strand NGS, so on, Mass Profiler Pro, all of these came from Strand. So in terms of impact and translational impact, the impact was in research. Then it was really interesting that I think we took a decision to pivot the company. That's a business term, but essentially to change tack from being directed towards research to being directed towards clinical diagnostics. And the tools that we had developed actually positioned us to be the first company in India to set up CAP-accredited NGS capabilities. So we started doing genome sequencing and so on for clinical applications. And that journey has led us to a point today where we are, I guess, 800 plus employees, about 40 PhDs in the company, 20 labs around the country. We do about 1.5 million tests a year. So it's actually been a long journey, lots of challenges along the way. I can at least remember 3 situations where we thought we'd crash and burn, but you know, sort of like a phoenix rose from the ashes. But it's a complicated process. But we didn't have incubators, we didn't have any support, essentially from the academic institution. I think that picture has vastly changed, and you will hear more about it. So I think it's a lot easier today to think about translation and taking it forward, and I think that's my message.
Absolutely, thanks. And if I may add, the journey that Chandru has just mentioned, I think many things have been written about it, but I've had a chance to look at it closely. Even the collaborations that you had with the UCSF-based startup 100X, which was a phenomenal thing in terms of the image data analysis and the software to allow building things which for research perspective, and then your, if I'm not wrong, Agilent collaborations, and now going to personalized medicine all the way, working with Tresta Labs from the HCG and other collaborations that you have.
So actually the work with UCSF ended up at Institute Curie.
Institute Curie, fantastic.
Right, for doing all the microscopy.
Fantastic, yeah, absolutely. Thanks for those inputs. Well, if I may request, Asim, introduce yourself briefly, and what do you like largely to talk about your journey from being a fellow to incubation center turned entrepreneur now. So if you can say that.
It's very humbling to be here, especially since being a Wellcome Trust first batch early career fellow. And I very fondly remember those 4.5 years of the fellowship, because it was very different. The way the Alliance runs is very different from how any other funding program in the country runs. So I started the company Prante almost just immediately after my fellowship got over, and I moved back to my home state, Bhubaneswar. Very frankly, I had no clue why I started the company and what I really wanted to do in the company. The first thought, or the reason why I started the company, because I was a peptide chemist, so I could make peptides, and I thought, you could make peptides in cell because nobody does that good synthesis in India. Very quickly I realized that's not how it's going to run. Fortunately, I was exposed to one of the programs by IIT Delhi conducted in collaboration with Harvard and SAI. It was a 3-day stint, and the first time when we got to think about what we could probably do for the company. Okay, and it was a small competition. And the inspiration actually arose from our personal experience. I and my wife, who was also my PhD colleague, she had a condition, pregnancy condition, preeclampsia. It during the pregnancy. And for about 4 years since then, we were completely devastated and trying to cope up with that. And when the opportunity came, we thought, I think this is the best motivation to do something. And when we started looking at it, we found neither India nor even globally preeclampsia is addressed the way it should be addressed. There are practically no diagnostics for this, and you really couldn't predict that you are on the verge of entering a complication. And that's how we started this company. It's been 4 years, so I was running my parallel academic career post my Wellcome Trust, then moved over to being a CTO of one of the incubation centers that runs the BIRAC Bionest, and had a tremendous experience watching startups, okay, from the very initial stage of idea to what all happens in that space. So fortunate for me, I was lucky to have a crash course of early entrepreneurship. Some I learned the hard way by experiencing myself, and some things I learned by watching. So I think that's a unique experience that the incubation ecosystem has brought into the country. And 4 years now, as I stand, I moved full-time into the company last month. And we have not only matured from preeclampsia being one of the targets that we are pursuing, but evolving platform technologies. Simply because if you see in India, there are many companies, but nobody makes devices to a level that international companies have reached. Okay, and not that because we cannot make it, simply because we haven't started doing it yet. So when Taslim shows you the funnel, the hypothesis is you put a lot over the top of the funnel and expect lot to translate into research. But there's an alternate approach to it. Why don't you just widen the neck of the funnel? So you need a lot more people at that neck who can help translate the exciting research that's happening into products. And that requires a slight change of perspective. Not that entrepreneurship is very different from doing research. It's just a new perspective, or you need to ask new questions. And if you could do good research, why can't you run a good company? I really don't see a lot of difference in the way you do things there. So that's what I have to say.
Thanks, Asim. So I think if I may take your last point, taking the funnel analogy further. So what you're possibly suggesting is that would the funnel, which actually have the neck come in, before that, can we actually have a funnel which has 2 layers in it, or a kind of a lower layer before, which could be seen as another next level of playing ground for translation further, and not necessarily the same funnel continuing within that you're mixing, and you're not going at top, you're coming on bottom, and so on. And that's possibly a zone that one could look at. Thanks, Asim. And now I'll go to Dipanvita, who I am sure has a perspective from a completely different, where she has looked at science for a few years, where she has been in the model of funding science in the entrepreneurship level as an equity model through the India Innovation Fund, if I'm not wrong. Then you have had the IKP and now IKP Eden and so on, and I'm sure you have seen very, very exciting academic discoveries going forward in terms of entrepreneurship or licensing and so on. So your views on that, Dipanvita.
So I am the chairman and CEO of a research park and incubator in Hyderabad. It's headquartered in Hyderabad. And I say it's an incubator and science park because it also houses large companies as large as DuPont and US Pharmacopoeia, which actually creates an ecosystem. We are not part of an academic setup. We are in Hyderabad, so there are a large number of research institutions and universities. But at the time when this was set up, it was set up by ICICI, and the government of Andhra Pradesh, and then Andhra Pradesh, gave us the land at the 200-acre campus. It was discussed that the research that's happening in most academic institutions, and I have to tell you that Dr. Mashelkar was one of the founders of founder directors of IKP, along with Mr. Narayanan Vagul, who was the then chairman of ICICI, and Ashok Ganguly, who was the chairman of Hindustan Livers. It was decided that the research that's happening is not translatable and industry will not be able to actually benefit from it. A new setup needs to be created in India which is not there, and if we put ourselves in one of the academic institutions, and this came from a CSIR lab, not CSIR lab head, DG CSIR, said you should be equidistant from various things because the business culture that has to be built in an ecosystem is not available, and I'm talking of 99, 2000, in any of our research centers. And that's what I hear from Vijay is very similar, because even the word business and word commercialization was kind of a taboo. Whether it is now is something we need to ask, but I think we have progressed a lot, and what has happened is because of various schemes like BIRAC coming in, which is a 7-year-old company, it's a PSC, from the Department of Biotechnology, which helps connect industry and academia through various schemes. And we, IKP, CCAMP, and many other incubators across India, several other incubators across India are partners, active partners of BIRAC. And I think that itself speaks a lot about the way government has been thinking. It is not like top-down, everything being governed from Delhi. It's about how do you partner with various local ecosystems, local bodies who have the pulse much better of what is happening in the ground, and work to see how commercialization can happen. It's been a fantastic, this is our 20th year. On an average, I see more than 1,000 proposals coming in a year. So I have probably read about 5,000 proposals, interacted with more than 7,000 academicians, I think 8,000, because we just look at those numbers. These are huge numbers. But I still think there's a lot to be done in kind of understanding how commercialization happens. And probably there's a philosophical difference between when a researcher starts working to explore from inquiry-driven science, inquiry-driven questions, to solutions-driven questions. I think there is a fundamental difference in the way it is thought, and how do we structure everyone cannot do and should not become entrepreneurs. I think we need good scientists, we need science to remain there, but is there a mechanism, and Vijay, you would know it very well, what kind of mechanisms, I think we need to develop these. What mechanisms should be put in that when there is something, somebody has to recognize it, acknowledge it, put a pathway for commercialization. Do we have those bodies? And we can discuss this further, because I think that the introduction remains short and we can.
Absolutely. No, thanks, Deepan. I think precisely, I think I think once we have set the context and introductions, the idea was to, dive into the challenges that we see and how possibly some of those challenges have overcome in different contexts and geographies, and whether those challenges remain the same and whether those require the same solutions or we require different solutions and different mechanisms and different. I think idea would be to discuss those today, and I think it's a good segue for me to get into the second stage of the panel discussion, where so if you look at it, a lot of if you read a book called Cambridge Phenomena, the author has gone very deep into illustrating how a 900 years old university, which was doing phenomenal science, has changed or rather transformed itself into what you call it as the innovation fountain in terms of building those innovations of different sorts and so on, without at all compromising on their basic tenet of doing fantastic science and how they have done it. And it is not possibly a solution, but it is a multiple approaches, few approaches gone bunk and few approaches working well. And I think we need to possibly be bold in terms of taking many, many approaches to support science going to the next level. But I want to start with challenges first, and if I keep the same order and start with Chandru, sorry, I should keep this. Your thoughts, and Chandru, you have seen different geographies yourself, Boston, Bangalore, of course, but what are the challenges traditionally which have been there and where they have come up? And let's look at the challenges for now and in India for now. What do you think where are those challenges? And then next round we'll come back to possible models, including the role of incubation centers as a next layer of the panel discussion. Yeah.
Sure. I think can speak about challenges both from just the perspective of the translational researcher who's then becoming an entrepreneur and so on, and also from the perspective of the institution. Right? So I think from the perspective of the entrepreneur or the translational researcher, I would say that you talked about Cambridge. I think the book by Vyakarnam and Fatke is very interesting. It's called Camels, Tigers, and Unicorns, right? And so what I found very interesting in that is the way they've laid out the challenges. And they're actually laid out what they call the three chasms, right? That you have to cross, the chasm, however you pronounce that. So the first comes up essentially when you're taking an idea to a proof of concept. The second comes up when you have to take the proof of concept to a product or a solution of some kind. And the third is, of course, when you're trying to scale product. And that at each of those, challenges, you need mechanisms to get you past, right? I think today what has happened is, particularly in the life sciences, you've seen an extraordinary push in getting an idea to a proof of concept. And there's a lot of seed funding available. So that challenge to me seems to have been adequately addressed. Right? And the state has played an extraordinary role. Not so much the private capital, right? Mm-hmm. The private capital, there are a few incubators, but they've really sort of focused on medtech and devices and health and spaces. The second challenge comes when you have your proof of concept and you want to go to a product, and that's where you need early venture capital, risk capital, and so on. And that has not been forthcoming, as far as I can see. We don't really have adequate risk capital available in the country. There are some moves to create things like the Bharat Innovation Fund and so on, which can take you to the next step. But I think the mindset of the funds are that they look at a fairly low risk, quick return kind of investments, and most of our life science companies will not qualify.
Yeah.
And then the third, is the jump up from venture capital to private equity. And for that, you have to have a proven market share, a product that has, been accepted by the market. You have enough paying customers and so on. And the financials are such that there's a large enough market opportunity that private capital will get in. And private capital will typically look for an exit within 3 to 5 years. So you really need something that's very, very ready to scale for that challenge card. Now, I think I'll
Yeah.
just wrap it up there.
No.
But I think, you know, these are some of the real challenges. From an institutional point of view, I think, you know, the challenges that we saw were that, you know, there was no ecosystem, and I think that has changed. I remember when we started Strand, SID and the director, Govardhan Mehta at that time, said that in the next 10 years, if there are 5 more companies, we'd be quite happy, right, that come out. Today, I just checked yesterday with SID, there are 20 translational companies that are currently operating in the campus. And a lot of them, like Patshod and so on, that Navkan Bhatt and others have developed, are actually doing quite well. And I think these are, so there's been a momentum in that direction. My final point is that all of us have to be prepared to be camels. If you look at the camels, tigers, and unicorns, we can't be tigers because we can't raise the kind of capital you need to be a tiger. And you can't be unicorns because God knows what that is. In life sciences, I haven't seen one yet, other than Theranos. But,
Let's not get
That was very
but so we all have to learn to be camels, learn how to earn also as we get started, you know, store some energy to keep going for the long run. And so.
Thanks, Vijay. If I may pick, I just wanted to pick on the thought. So, you know, when one looks at the journey of science further, what I would like to add is that there's definitely when one talks about commercialization, the biggest word that joins it is entrepreneurship. But I would like to add to that is that there are other opportunities to take the science forward. It's not that one needs to possibly become a CEO or a CSO of the company that you form, but there are different forms of taking science further. And I just wanted to add to that discussion that where, where there could be an individual, because I think the participants here, our colleagues here, are early career fellows, mid-career fellows, the senior fellows who are leading their groups, leading the fantastic science. The opportunities are there in entrepreneurship. I think Dipanita clearly mentioned about the funding opportunity, which is there. Phenomenal funding. If you want to try your idea, there is a grant available to test. If it works, good, set up your company. If it doesn't work, continue the science that you're doing. Many places do not have that. Most geographies do not have that. Coming back, but can that be a translation which is not directly linked and not necessarily be an entrepreneurship route, but can be of taking that from the TRL level 3 to TRL level 7, where an opportunity for that could be for collaborating with a startup, a biotech, or an industry to take it to the next level. Because that is another way where you can actually say, I do want to take this science forward. I definitely am very, very excited on seeing the impact of my science going to the market, right? Having impact young children, mothers as you talked about, and other people who are impacted with other healthcare, non-healthcare challenges. How does that route look like? Like entrepreneurship is very, very exciting because actually you then take the bull by the horns and you actually say, how do you want to do that? But here, how does that look like in terms of, if you were not to set up a company? Was there a chance for you to do while being in ISC and do that?
Oh, yes, there was, right?
Sorry if it's a curveball or something, but please.
No, in 1994, a bunch of us actually set up the first 3D printing facility at ISC. Gurumurthy, Swami Manohar and I, we pooled our monies and grant monies, and we bought a Stratasys 3D printer, and we used it for printing various engineering designs, which could then be used to make molds for manufacturing and so on. And so Manohar and I worked more on the geometric modeling and the computational aspects of 3D printing. And Guru ran the printing center in the mechanical engineering department. And that was the first 3D printing and rapid prototyping facility in the country. And it was, it was a great attraction. We started getting companies, the automobile companies, and so on, showing up at our doorstep saying that they wanted designs to be printed and so on. So we were not ready to take it up as entrepreneurs, but we had a conversation with Tata Consulting Engineers, and they came in and set up an organization called APDAP. And APDAP became sort of an outreach to industry in Bangalore, in and around Bangalore, to actually provide these rapid prototyping services. And today that APDAP has sort of evolved into CPDM at IISc, the Center for Product Design Manufacturing, which actually also offers academic programs which are very driven towards translation of designs into manufacturing. So that was an experience. From within Strand as well, we did some research that we were not ready to take forward. For example, we developed the virtual liver platform, which could predict hepatotoxicities, both with an in vitro and in silico platform, but could give you in vivo sort of predictions. But to commercialize it, we needed a CRO arm, which we weren't ready to build. So we sold the entire patent and the team and the lab to Syngene, and it now sits inside Syngene International. So sometimes
Yes.
you even do it from within a company.
No, absolutely fantastic. Thank you. Asim, when the transition for you has been, if I may say so, quick, in terms of you have looked at three forms of science being done in different flavors of science being done in terms of supported role and so on. Through these transitions, would you be able to identify and possibly share some of the challenges that you see that happens and exist in our ecosystem? And if a couple of them you can just mention before we, in terms of that you saw that if this was there, because of this I could move forward or something that range. Not possibly for you, but in general.
Yeah. So I'd probably introduce one of the concepts. So we talked about TRL, okay, Technology Readiness Level. But when you're talking about something going to commercial, as a commercial technology, there is another scale which is called as the BRL, which is the Business Readiness Level. And by default for all deep tech based ventures, BRLs always lag behind the TRLs. So if you are at TRL 7 or 8, your BRL would probably be around 2 or 3. Now, my understanding, since I transitioned relatively very quick, as you said, but I was exposed to this idea way ago in 2007 through DBT's first entrepreneurship program, which was the YES competition, which now is runs as best every year. And we are exposed to the concept of commercial technology way back there when we were PhD students. But trust me, I at that point of time, I never thought I would start a company any day. So let me just introduce this concept very easily, simply. If as an entrepreneur or not as an entrepreneur, maybe simply as an academician, I look forward to a technology being a product where a consumer pays for this product. I need to be on the two boats at the same point of time. While I am trying to push the TRL levels, I also need to look at a compatibility of the BRL levels. Most often we take the paradigm that let's go sequentially, let's reach TRL 9 and then we'll worry about BRL. And that's, I think, is a wrong way to do it. Now, so when you kind of go to BRL 9, there is a stage, you come as the ideation stage, then you come to the prototype stage, and then you come to an advanced prototype stage, and then you call something as a minimum viable product. The unfortunate scenario is we really do not have clear definitions of what is a prototype, what is an advanced prototype, or what is a minimum viable product. And just like anyone's life, you really can't define what you will be at 40 or what you will be at 25 or what you will be at 50. So we miss looking at the BRLs. And the caveat that you fall in if you do so is that while journeying to TRL 9, you could have spun out some simple technologies which are low-hanging fruits, simple, maybe not phenomenal in commercial sense, but still has few takers. The reason to go in this way is that if you are trying to do good business or really make a technology that has good social impact, because that's what we scientists, even if we turn as businessmen, that's what we are looking for. Then you also need to understand how the business world runs, how the market runs, and how a consumer thinks. And for every stage of the technology, there is a time. Just like we heard yesterday, Dr. Bhang saying, the time is a very crucial factor. Sometimes your TRL 9 technology is advanced 20 years from now. You know it's good tech, you know it will survive 20 years, but as of today, there is no one willing to pay a dime for it. So what sense does it make? And maybe 20 years down the line, even though you were the first mover, you had the advantage, you can never capitalize on that advantage. So, the second mover or maybe the third mover or maybe the fourth mover is the one who is actually capitalizing on the tech or the hard work that you built, which was so futuristic. So, my sense, or rather the challenge I see, is that we need to train ourselves even as researchers while we are doing it to look at TRLs and BRLs at the same point of time and see what comes out of it at various stages. Let's make it sequential, not by the chart, but sequential from the perspective of a product.
Thank you so much. Aseem, if I may just reflect, I think, and we will come back to on the third point, but would that possibly translate into a saying that, when a PI builds a lab and wants to take it forward, they actually want to be in an institution, in surroundings which can nurture it more, right? Where it's not about your 200, 300, 400, 500 square feet of lab space with fantastic facilities, but also the surroundings, the people and so on, which can add intangible, but absolutely invaluable additions to your thinking, your thought process to take your science forward. When one is building a translation approach, does it mean that you actually should also look to engage with an environment which has possibly the mindset and some of the bearings of this aspect, be it with the people who talk about it, to actually have that value add rather than continuing? And that could be a very interesting model of taking the translation forward. And while we'll come back to it, I had to say it, that incubation centers in and around institutions actually provide that environment to not necessarily only let the science happen in the lab, but also when doing translation, can that be engaged with an environment which can give insights like this? Not TRL to BRL, but also how does it look like, what would industry want, what are the investors want, how does the market look like? Because when it goes from a curiosity-driven attempt to a solution-driven attempt, the users actually change, and it matters a lot. And who is going to support in your journey to build solution for a user are completely different players, and they play on a nearby looking soccer field, but different soccer field in terms of the grass, in terms of the other aspects. And that engagement possibly is important and valuable to do that. And I think that model would be also a good model to look at. I'm sorry I had to intervene, but I thought it was a good built into it. Now, while it is important to look at, I'll ask Deepavita, in your, and we are continuing, but if you can begin with the challenges that you saw. I think you mentioned Dr. Mashelkar indicating that the research is not translatable. If please
20
correct.
years back.
20 years back, 2000-ish. Have you seen something changing? What are the current challenges that you still think remain in the broader sense?
Yeah, actually the environment is changing. No environment is static. So it is dynamic, it is following where the world is moving. But what happens is, typically a researcher, a serious scientist, has an urge to share the science and publish, which is absolutely normal and what it should be. Who is going to actually tell the scientist that yes, there is a requirement for this, there is a possibility, what you have done is unique in some way which can be used and commercialized as a technology or a product can be built around that. Is there a mechanism or does the onus lie on the scientist? Can a scientist who's been trained in science and not in business, or not even looking at technologies, to do so? Or should there be other offices like the tech transfer offices that normally exist in most universities and research centers, and capable people going through the science and saying, no, this needs to be patented, this is a know-how that should not be patented, or this is okay, please publish. So that mechanism, I think one needs to really see how that's developed. So, you know, even if I'm talking of large pharma today, and I do not know the mix of what kind of life sciences you do, the people in this room do, but if I say drug development, the latest report that I saw says that anyway 90% of the candidates that have passed through talks fail in clinical trials. 75% of the products that go to the market do not justify the spend on research. That means 25% justify. So, and that 25% gives all the revenues to the pharma companies. And by 2020, the net ROI of the net investment that you expect, the return on the investment that you expect from your research will be 0. If this is from a large venture capital company in the US, it's a Forbes report, recent report, and this is from the large pharma perspective. If that is so, if we do not know what they are doing, what their pathway, what their molecules are, what is their pipeline, even if we are working on something, how would we even think that this is translatable? So where is that interaction? Where is that knowledge sharing? Where is that complete grasp of saying what is happening globally? Where is the market moving? You know, and with a very, very interesting data, and I'm sure you know, all pharma are largely into deep data now. And there are in drug discovery, drug development, globally there are only 125 companies that have raised money, reasonable amount, not significant, no unicorns, significant amounts of money. In healthcare there are 1,000, and must be Strand is one of them. In drug discovery, and actually Easy is one, AstraZeneca is one of them. So these companies are working extremely seriously on looking at how AI is going to impact, and what is it, how will they find that magic pill to crack the stagnation of their pipeline. In this kind of environment, what is the path that our research centers should take and what would be the approach. I somehow feel that yes, the incubators have extremely important role to play. One, because the incubators don't do the science. The incubators follow what's happening and probably connect to the industry and people who are on the other side of it, are interested in seeing the products go into the market. And so there has to be somebody, some element, some sieve, some organizational entity saying that these can be translated, sorry, these cannot be, fine, you do the science, but these are the priority areas for an institution to take forward. And I've interacted with several scientists who say our TTOs are not good. Actually, I think it's a national job to first create the right tech transfer professionals and tech transfer offices, because if we do not have professional tech transfer people in the country who can really help the scientists, guide the scientists, we will not see even if the increase of your pipe is basically wrapping it around professionals who understand what is to be done from this side of the sieve to the other side of the sieve. And I'll stop at this and
yeah, sure. Thank you. No, thanks, Ibrahim. I think one important aspect, and I think which was being looked at in terms of engagement, and I remember an effort at CSIR called CSIR Tech, which aimed to build that aspect, and I think possibly different models of the same are required, and more so, but glued into the organizations, with some sort of porosity that we have to build in to do that. That would be much more, because if they remain to be, and this is my opinion, so I'm happy to be corrected and being proven wrong, but if they remain to be the business sides of thing and not the science-taking business side of thing, the disconnect actually results into the non-productive outcomes than otherwise. So I think if they more talk the science, which is going to go business side, but rather only business side, that would not. This is my opinion on that, my personal opinion. So I think we have heard a few things. I'm very of time. We have around 12 minutes or so. While the third point was being there to bring back on the incubation, but I think we have covered that aspect, and I think we'll let interactions cover that more of it. But just to give, I think it's very important that I do think that this, India Alliance possibly identifies top-notch fellows. I'll not go further than that. I think this is a phenomenal gathering, and I think it would be fantastic to have a conversation in terms of how to, take your discoveries and science forward. And this panel possibly would be able to add some perspectives if you have some questions. So please go ahead and ask questions in no particular order. If I may just, I think if the mic is a problem, just shout out and we can build from there. So I'm looking for some questions before we start.
Usha Menon. I want to just take a point that you just made. Take up a point that you made. So you might have a very good tech transfer office even. So just yesterday I got a paper returned saying that because the business office told us not to reveal some coefficients, the paper is good, but they are not willing to publish. So of course, for the mathematicians in the group, they're like, just reveal it, because the fellow needs to go for the next fellowship. We need that paper out. And I find that's always tension, and whether this will ever become a product so far down the line, but what we can gain by just revealing everything is so tangible that we'll get a high impact paper, and that fellow it'll definitely make a difference to his fellowship application. So I'd like your thoughts on how you do that balance.
I would think, and we can debate on this, my personal opinion is it should be left to the scientist. If a scientist feels the urge to take it forward, I think it is the right of the team and the scientists to publish if they think that they do not want to patent.
No, no, so it's not about, they're just trying to decide between, so they want to do this thing.
Yeah.
But the
They're
predators
okay going the other. There are many scientists who say, even if it is patentable, I do not want to patent. And I think it should be their right to be able to say so and say,
So
I
that's a
want this as
different-
public good. It's
So
fine.
that's a different issue, because that's a straight black and white.
Yeah.
I'm not talking about the black and white because it's really easy to make that decision. So I'm talking where you can see there's some value down the line. You're right in the beginning. But the tangible, it's like eating now and getting fat later, all those
So
things.
who decides? I think these are all subjective, but if there are professionals who have seen many of these going forward, would be able to take a little more informed decision than
I think this
a
goes
scientist
back
does.
to the idea that you need to have a really professional, good tech transfer office, right? Because these evaluations can be done very quickly if the right people are there in the tech transfer office. The Harvard Medical School, for example, a couple of my classmates from MIT have served as the tech transfer officers, and they were PhDs. They become entrepreneurs, worked in industry, then come back, and they were all guys who could be. You could go grab them, go have a cup of coffee, sound out and tell them about your research and your paper, and they would quickly advise whether or not you should hold back or take it forward. So these are the kinds of support services that I think Deepamrita is talking about when you want the researchers to be able to resolve these quandaries.
I think, if I may just add, I think absolutely, and while those, I think I would say that till that happens, the places where such dialogues happen is a very important aspect, as while tech transfer
And that is
offices
the
come up,
incubators.
absolutely, because while they would come up in time and in shape that would work, I think the current, let's say if somebody from this group is attempting to do that, so my colleagues on campus from InSTEM, one is actually building a fantastic work on a cream, an ointment which would protect farmers or farming laborers from the effect of the pesticides, which is absolutely toxic. And that work, it was an easy thing in terms of understand whether there is a market for it, whether there is a need for it, and then how do you plan it? Like, if it is a know-how or a trademark base, that's a separate thing, but do you want to patent it before you publish? The paper has come out well. I think it has been ranked by, of course, several people, but when Hindu covered the last year's top science being done, it came #1, while it was done clearly from the perspective of doing a solution base for the agriculture, pesticide-related toxicity. So I think tech transfer offices, incubation centers, and possibly other aspects, it's engagement, if I may call it. It's an engagement that has to happen more often. And the more you engage with people which possibly talk about other aspects of science taking further. I could not put the slides, but I think there is a fantastic article on accidental discoveries, 8 accidental discoveries or innovations which changed the world. When the idea was to build this, but during discussions it could do this, and we know a lot about that, and that also come out in that aspect, whether this could work for that as well. And that is the engagement possibly. So if may call it, I think it's like anything else, it's engagement and exposure. It's the key to do that. And if the environment is created by incubation centers, tech transfer offices, and other things, that's a boon that it would help.
So I'd like to just put a very small word. What you need to have is a person with skills in strategy. It's as simple as that. And a strategist is not a person who has studied strategy. A strategist is a person who can align your desire from whatever tech you are developing, your vision, with every other variable in the existing in the market at that point of time, the next 10 years, and create a roadmap which you should follow for that one invention. And strategy is, you know, it comes innately to a lot of people. There are ways to study strategy, but you know, when you call as a tech transfer officer, a good tech transfer officer is a person who can design a good strategy for you.
Anurag?
If I can just, go back to this point of tech transfer officers, how does one train to be a tech transfer officer? How does one build a tech transfer office? Are there any national level programs for this? So from an institutional perspective, if one wants to set this up, how does one go about it?
Actually, there's also an association, it's called STEM, S-T-E-M, Society for Technology, whatever, managers. So I'm part of it, I'm on the board, but you know, there are several very well-trained, globally acclaimed officers like Autumn, AUTM. We can get Autumn to do the training. I think it's the intent and the will that is important. Then, for example, Oxford Innovations, they do fantastic, and it is about learning the process, and scientists have to get into this business, and science management is weak in India, and they think we need to build science managers. So this is also a kind of a science management process, and it's a career, and that has to be done.
So
and I think
I'll just add possibly, so DBT has a program, if I'm not wrong,
yes,
which people, there are, yes, possibly, where they take people to Wisconsin for training, for especially for tech transfer and so on, for 10 days every year, if I'm not wrong. There has been an attempt, and I think there's need to be done more possibly to address that aspect. Yeah.
much more.
There's a question from Shah.
Very interesting discussion. We've heard some really inspiring case studies, and case studies are always interesting. I'm an epidemiologist, and it always seems to me it's important to get an overview, to get the meta picture. And what we know globally is that 90% of tech startups fail. And the problem with failures is that nobody wants to talk about them. But failures are actually they're jewels because they help us understand how we should proceed in the future. University tech setups in the US, in the UK, have a similar 90% failure rate, but vice chancellors don't want to talk about the failures. They get buried, and the academics often get buried and have to find jobs in other universities. In the UK, we've invested something like £300 million over the last 2 or 3 years in setting up incubation sites in British universities. The first audit of this scheme has just been published, and it shows some really interesting findings. We should have put all the money into Imperial College. They're incredibly successful. Oxford and Cambridge are not far behind, but the other universities have done virtually nothing with the money in terms of return. In exactly the same way as with the pharma companies, the return on the investment is about evens with what comes out. And what I would suggest here is that we've got great opportunities to actually set up systems where we can audit, we can follow up, we can survey, and actually get information of what was started, what finished, what was successful, and learn the lessons. And we've been doing this in the US and the UK for the last 2 decades, encouraging universities to get involved. And it's taken us 20 years to get to the point where we're beginning to get a grip on what's actually happening. And so in an environment where things are already happening and have been happening for the same 20 years, we've still not got an Indian survey of startups in biotech in the universities. And it's about time we have the survey so that we can actually get a handle on where we should be going.
Couldn't agree more. I think about 4 years back, we actually wrote the blueprint for the Atal Innovation Mission of the NITI Aayog. About 4 of us got together and drafted that. And now, at that time, we interviewed all the incubators the major heads of incubators in the country. And we kept asking for audits. And there were no audits, right? And even till this day, the numbers put out are, the amount of money distributed, the number of startups, but it's never about, what happened beyond that. And so, in fact, in our report, if you go back and read it, it's on the website, we actually say that don't put money into incubators blindly, right? Because, we are not sure at all of how they perform until these audits are completed. And we said emphasize more on grand challenge problems because there you can track much more closely. But that's not how it evolved. If you see what the NITI Aayog just went ahead and did, it was very different.
I think Anurag has a question for
Yeah, partly a question, partly a comment, because you mentioned, for example, that in response to Usha's question, that one must leave it to the scientist in terms of whether they want to commercialize or not. That too gets a bit complicated, especially in institutions like CSR that have a mandate towards commercialization. So in our institute, we are following the policy of CSIR, in which before a paper goes for publication, two scientists or the institute need to review the paper and say, is there any potentially valuable IP material in the paper? If the answer is yes, it doesn't mean the person cannot publish. It means steps must be taken to file a provisional patent and then move on towards capturing it, the additional money, and then the paper can go ahead. If the answer is no, then there are two people, other signatories to the process, that there is no value in the paper. I'm not trying to say it is necessarily the right thing to do, but individual goals and institutional goals and national goals are sometimes a bit discordant. And one must be cognizant of that, and not all power sometimes can be directly given, please publish. But the student would certainly like it to be published the fastest possible so they can move on with their career for a postdoc and so on. But the funders do also have some right over what needs to be done. And in a very, very personal context, I realized this when we had a very nice paper in review on a particular algorithm that by itself was potentially not patentable. It's a mathematical algorithm for judging lung function at the very high sensitivity, maybe 10x compared to current methods. And we effectively almost realized that we're going to pull that paper out, and we have not published it yet. It's been 4 years. Because we competed for $1.5 million startup funding from USI Tech Endowment Funds, built our own device along with Rice University spin-off. It's gone for CE certification, should hit the market next year. Because the long-term returns of building your own indigenous device with that extra knowledge to give you the edge in the market was worth more than getting that paper out. But sometimes you're in a position where you can make these calls, and I would not fault anybody who could not make that calls, but institutions need to make a policy which is beyond the individual scientist. So I think you would probably agree with me, but I still want to see your perspective.
Yeah, only thing one has to, it's not about trusting, it has to be convincing.
Absolutely.
So consensus is built, like what, Bala, Dr. Balamanian, who sits on a board, Bala keeps saying, Dipanita, either there has to be consensus, either you convince or get convinced. There cannot be that you walk out of the room. So you have to have the power to convince. So I think that would be
Indeed. Absolutely. I think we have, hit the time buzzer, but I think if I may just say, there is a phenomenal opportunity actually being built right in this ecosystem in India. As Vijay says that when he started out, there were no support system and so on. So while the funding agencies which are being on a high alert to support those translation ideas, so while there are entrepreneurship funds, but there are also translation support like CRS that they gave, the PACE they gave, and there are other supports they gave for only translation aspect. Then there is a major, environment of incubation centers are being created. We at CCAM, we have an incubator, and I think if I may say it from that experience, if there is a lot of knowledge that is being generated, and I think it would be a fantastic idea for CCAM to actually reach out and make that knowledge being available, but also a phenomenal scientists like you to actually engage with incubation centers like this, because it's absolutely fantastic that what thought process and what discussions go on there, which can actually shape, what Anurag rightly said, your decision in terms of going forward and not. And I think those processes possibly need to be put in much more, so it is not a well-intended environment, but rather process-driven environment, so that nothing doesn't fall through the crack. But I think I feel very, very optimistic, and I think I'm sure my co-panelists as well, because this is where the science starts. And there is always an importance in terms of, say, taking science to the forward. Not all of it, but there's a lot of it. And I think the more we do it, the more we discuss, the more process we put it, I think the end product will be something that we all will be proud of it. I think with this note, please join me to thank my co-panelists, and thank you so much for having me.
Thank you, Tasneem, and thank you, panelists, for this really interesting and thought-provoking discussion. With this, we will break for tea, coffee, and request you all to please be back in 15 minutes for the next session. Thank you.
at NIMHANS. Over to you, Akhilesh.
Thank you. It's a pleasure to be here and talk about where we stand at the end of 3 years. And I just wanted to talk a little bit about why is it called Center for Molecular Medicine. It goes all the way back to 1987 when I was 1 year away from graduating from medical school at AFMC Pune. And what I realized was we were doing anecdotal medicine and we were doing presumptive diagnosis of tuberculosis, malaria, diarrhea, dysentery. And my mind was not that type. I liked objective evaluation where I needed to be sure. And luckily, one of my classmates convinced me that, have you thought about doing a PhD? And I said, okay, that meant my dad selling the first Marutis ever sold in the country, and I got out of Army, although I was a hardcore Army types, and then I went to Ann Arbor, Michigan to get a PhD. But this idea was that the future of medicine would be molecular. We need to understand what happens, why it happens, for us to make a bigger impact. So here, through this, I got an opportunity to try to say how our future efforts, if the future of medicine is molecular, or at least one of the futures of medicine is molecular, how are we going to contribute to this? We need to build that as a system. And I have to start with thanks, because Margdarshi program had just changed some of his guidelines. And I read them, and I read them over and over again, and I was still stuck because I had two major problems. One is I was in the US, and I was going to stay there. I was at Johns Hopkins until very recently. For 16 years, I was there, and I was going to stay there and yet spend part of my time here in India. And the other thing is I had already started many years ago a non-profit research institute which has about 50 people in Bangalore called Institute of Bioinformatics, which is a systems biology institute. And I thought it would be taken against me. So one seminal meeting with Shahid Jamil, and I asked him, do you think I'm eligible to apply? And he said, you never know, but you should. He encouraged me to apply. And I think I had the toughest interview with the panel, and I thought this was the end of the world. And then, I said, okay, that's fine. I don't get it. My family gains 25% of my time. They will be happy because they were not going to go back and forth. I come back and forth. But then as it would have it, I got it. I'm very thankful to the committee that they placed their trust. And what I want to show you is how we are walking towards establishing that Center for Molecular Medicine. So I want to also thank all of the staff, and they have been very, very helpful. I've been brought to tears many different times, but I also want to tell you the chronology. So in July of 2016, I was funded. The money was transferred, more than $1 million sitting in the bank. For 6 months, I took a sabbatical from Johns Hopkins. I came to Bangalore, and I was sitting there. I have never had this much time where I was twiddling my thumbs doing nothing. And that brought me to tears. So what you may or may not know is Shahid Jamil also does psychotherapy. So I had to call him and I had to say, how do you help me out? So life went on, and I thought I knew about India. I grew up here. I knew the system. I had operated back and forth. What I didn't know is how many of the institutions work, where if you order a chair, it has to, now I understood the real meaning of through proper channel. That chair has to be ordered through the director. How do the directors have time in the world to actually see a screw, a chair, and every single thing? So anyway, all this meant that 20 months elapsed. I kept writing my dummy updates because I had to tell chronology-wise, the lab is not operational yet. We are getting ready. We had hired people, but in February of 2018, all the instrumentation, including the mass spectrometer, it all arrived. So what you are seeing is a progress about 14 to 16 months. So you have to bear with me, and I hope you'll agree that we are well on our way to change the paradigm. And one of the important things for me was, in order to do this the most biomedically relevant research and translational research, you need to be in a hospital. It's a passion of mine. You heard from Dr. Bang, go to the people. But here, go to the patients, but also go to the cutting-edge physicians and others who are fighting every single day. You may have gone to medical school 20, 30 years ago, and even that medicine is changing. You need to know how people are being treated today. So I said, forget everything else. We need to be in a hospital, and we chose NIMHANS and across the street, Kidwai. So I would like to thank many of the people. So I like to again highlight outside the country, you all know this, but I still cannot get over the fact that the government of India is so generous. If you are almost any good, they will fund you. They have so many postdoctoral programs. They have so many programs for students who get PhDs in those labs. So Wellcome Trust provides funding, but there's also the flexibility. So we can hire some folks under the Wellcome Trust umbrella, but then use the money for consumables, because we have assembled a team of people who are doing molecular biology, proteomics, computational biology, molecular biology, microbiology, and I would especially like to point out 3 individuals here who are doing their PhDs. She's an MD in pathology, and these 2 are MD in biochemistry. She has done stem cell work, and she has done cancer biology. So all of these people are now sitting there. We have, because we are there, now we can say, Dr. Satish Girmaji, he's a psychiatrist, neurologist, microbiologist, neurologist, geneticist, cancer biologist, and microbiologist. Now we can go there and start to use the power. And I think what I want to highlight is we all say 1.2 billion people and all that, but I think the clinical acumen of the physicians in India, what they are doing in a few seconds and minutes, because the lines are so long, you cannot beat, and I'm going to show you how that plays back into the science. We also have many trainees on loan. So we are just trying to say, I think I'm not a business guy. These are some terms that business folks use, but the word leverage, I don't like it, but I'm going to use it now. We are trying to leverage whatever we can to take these funds and make them that. We want to transform the system. And the way we are doing that is, so NIMHANS has a program where they have MBBS and MDs who are doing their PhDs. So he was working with the psychiatrists. We take them over, and we start to work with them and make them do molecular things with us. So they are big bridges with the other clinicians. He is with a neurologist, Dr. Pal. She is with another neurologist. He is working in a genetics lab. So these are all MBBS PhDs. She's a PhD working in a microbiology lab, and he's also in a microbiology lab. So again, so we have a lot of firepower, but you need a place. So what I also learned is you have to fight with the procurement system and the government contractors and all of that. So I have also seen examples where people go to NCBS and they see how they should basically say, this is the model we want, let's do it at NIMHANS. Now, the way the system might work is you may not get what you accomplish. So we basically said, we also want to replicate NCBS and InSTEM-like ideas, and we have tried to build a lab that is somewhat like that. So many of these things, we wanted top-notch. And this is, we have the equipment funded by, all of this is funded by the Margdarshi. We have mass spectrometry, cell culture, computational stuff, molecular biology. But again, every lab at the end of the day is the same in every part of the world. It's just the people and how you do your science. So the reason I highlighted, I'm thankful to all of them. Now we are in the right setting. We can start to do science. And what I decided to do today was that I will not tell you how we are dissecting one protein, but how we are using this umbrella to try to do translational science. In my last slide, I may have to skip over many. I will tell you what are the other things that I won't talk about. I like to think our team as molecular detectives. We have made such major advances in research, we have technological platforms, but we are not training many of our folks, I think, to be molecular detectives. That's the term I use. You can say forensic analysis, because most of the time what we are looking at is we are working with things that we cannot see, touch, feel, smell, or hear. We are convinced they are there, a gene. It's a concept at the end of the day. You amplify it 1 billion times, you see a band on a gel. It's not the gene. It's an amplified chemical product. So we need to be molecular detectives here in the context of inherited genetic disorders. And again, some we have also meandered in that system. Some things take off, so we have started focusing more on those areas. And other things where we had obstacles, we had said, okay, let's school it. And I think you need to be agile in that way. People know agile programming. We have to follow agility in our life. And again, the India Alliance allows you to do that. So it's an old story. There is a syndrome which was intellectual disability. So this is work with Dr. Satish Girimaji. And it's a heterogeneous syndrome where intellectual disability is one part of it. Now, the reason that experience is so important is it was described 4 decades ago in a family of 3 males. It's X-linked. So you have to be a walking encyclopedia. When a kid walks in, they have all these features. You are basically comparing those features with everything else the textbook ever taught. Except that here, it has been reported in the literature only 6 times. Only 6 times. So you got to say and Meyer described this. 6 families have been shown it's that one. We cannot do this. Our students cannot do this. We relied on them, and what we are doing is now, okay, what can we do to figure out the molecular cause? So in this family, there were 2 male siblings. They both have it. Looks like X-linked. We are not sure. So this is clinical assays. Array CGH cytogenetics. I think we have to really say we're in the 21st century. Call a technique for what it is. It's a low-resolution method. We said, and again, I'm not saying what we are doing is earth-shattering. None of what I will talk about is earth-shattering. It's application in that setting, in that way, that makes it earth-shattering. Nothing, it's all routine. People will say it's being done everywhere, in the country and outside. Of course. So these are the 2. There's one unaffected. We do the sequencing. We figure out, okay, it is consistent with this. And here what we are talking about is that there is a deletion of 2 amino acids. And those 2 amino acids are very, very conserved in a gene that actually is an E3 ubiquitin ligase. A lot of people have done a lot of molecular biology on it. But just doing that molecular biology does not actually give us predictive power to say what will happen in a human. That's the truth. So exome sequencing says it's an unsolved mystery of 4 decades. It's an X-linked. It's a splice acceptor site. And as you can imagine, so it's a 1 base pair change, exon skipping, 2 base pairs deleted, 2 amino acids deleted. If you had exome sequencing data, you would ignore this. That's why you need to sit down together and build that team. But now this mutation, now that we have published this, it will help other people not ignore it. This is a big problem in all genomic analyses. So that is one case. The other one that I chose as an example is that there is a set of disorders where kids are born, and it's called Mendelian, meaning inherited susceptibility to mycobacterial diseases. And I'll come back to mycobacteria in a different context, but you've got Mycobacterium tuberculosis, but many other mycobacteria. What's happening is they're born with this immunodeficiency, and they have severe and recurrent infections caused by virulent mycobacteria, code word for Mycobacterium tuberculosis, but many others which are also mycobacteria. And you can imagine diagnosing these conditions may not be easy. Again, you need to work with experts. If you look at genetic basis, there are many genes that are known to cause it. Autosomal dominant, autosomal recessive, X-linked. All over the map. So genetics won't tell you just following the family. So this is a tragic story. 2 kids, you know, BCG vaccination. It's universal in India. You give the kid, and to piece it together may not be so easy because kids may die for all kinds of reasons. The clinicians have to interpret what the heck is happening. In this case, even though it happened 2 times, now people are on the alert. They may or may not know what is happening. 3rd child has some symptoms. Initially, look at this. This is still modern medicine. They are being treated for dermatitis because that's the symptom. Molecular basis will tell us where is the problem, what we have to do. They figured it out. We have a research project that is going on with this, and the 4th child. So here what we have done is this is interferon gamma receptor, has receptor 1, receptor 2, and both of them are known in this case, except the mutation that we find is this 1 base pair change. Again, it is in a splice acceptor site. You here there are 3 amino acids that are being deleted in the extracellular domain of this subunit of interferon gamma receptor. And that is tied very intimately into the biology of tuberculosis and other infections. But this mutation was not reported, and again, you would ignore it otherwise. So these 2 kids, so we have a happy ending. They have undergone a bone marrow transplant with their dad. And this is this particular form. We have a homozygous. After bone marrow transplant, you can see there's heterozygosity in the blood because now you have a bone marrow transplant. But I think from now on, anybody identifies these, it's very important for genetic counseling. We have just begun. Now, I think we have shown to the system and to our own people. I showed you the list of trainees. We are sitting in a gold mine. Although theoretically we know what all we can accomplish, but I think we still need to periodically brainwash ourselves or people we work with. So now our young folks are excited, including physician scientists. Now we have collected, again, these are world experts who have been doing this for a living. Luckily, they're collecting some samples, not all. 81 families with young onset Parkinson's disease. It's a craze to study to find the young onset. If you have a population of 100 million, not easy. 5 million, forget it. You got a large population, but well-trained clinicians, now you will get those. 5 families with familial Parkinson's, again not common. 4 families with late onset ataxia, 15 families with movement disorders with suspected mitochondrial problems, 5 families with familial and many sporadic neurodegeneration with brain iron accumulation. Again, very specific entities. They have been cataloged. They're sitting in the repository. We need the funds. But I think what's the most important part in all this is, and that's why I think outsourcing many of your analyses to some place where they'll just spit out something, whether or not they have one medical or clinical geneticist who can sign reports, is not the answer. The answer is we have to have an interdisciplinary team. People have molecular tumor boards. Those are becoming more established in academic medical centers. Not that most places still have them. Here you've got to have neurologists, neurosurgeons, psychiatrists, radiologists, geneticists, bioinformaticists who understand genes, proteins, and everything else that's going on in the patient. Now we can bang our heads to say it could be this, it may not be this. There are too many possibilities. It's easy to say exome sequencing. Actually, you're banging your head on every single case, so we need to have this. It's ready and in place so we can do all of this. Center for Molecular Medicine. I'll just change gears. One of the things across the street, gastric cancer. There was a paper that just came out by a team from Korea. They have looked at gastric cancer. They just published a cancer cell paper. They have looked at 100 different cancers. At the end of that, it was a national pride for them. It took them 10 years to do it. They spent many, many millions of dollars. At the end, I've hired the first author of that study. He's in my lab there now. They have accomplished nothing because we love to molecularly characterize and say, yeah, we just see observation. But the main thing is gastric cancer is a killer. And I told you, I'm very proud of having those physician scientists on board. One of them is this pathologist. What people are not paying much attention to is this entity, which looks very different under the microscope, intestinal type and diffuse type. When you dig into this, these are the 2 commonest subtypes of gastric cancer. One of them occurs at a young age, more common in women. The cells are not sticking to each other, and they infiltrate the stroma as single cells. That's what you saw. Poor prognosis. So one does much more poorly than the other. We do not know the molecular basis of this. Who is solving? When you go and look at TCGA, name the study, people are so proud of saying gastric adenocarcinoma if they will say that. But they are not looking at the molecular subtype, which for me is different. We need to think of them as different entities, and pathologists have always known this. But it's just that research is getting, in my mind, increasingly also disconnected with clinical medicine. We need to always be talking. Sometimes bioinformatics people take everything and run away without talking to the rest of the team. It's not just saying any clustering will give you 3 subgroups, but we need to be rational about it. So we have started exome sequencing, RNA sequencing, protein analysis, glycoprotein analysis, and kinome analysis of these samples. We have hundreds of samples. These are the early results. I won't bore you with the details, but I just wanted to show you that we have already found molecules, proteins in this case, by proteomics, which are upregulated only in intestinal subtype with no association in the literature to or only in the diffuse type. You could say there is no targeted therapy for these types. So they could become potential targets. We have also used chemical tricks to look at N-linked glycopeptides, and many of the tumor markers, as you know, ovarian cancer is a great example for that. They are sugars on proteins. So we are now saying we can do genome-wide. We can find thousands to tens of thousands of glycopeptides. And we want to look at who is more in one or the other. It could be either a hook that we can target on those cells, or it could be biologically driven. So this is to show that, yes, there are big changes in this case, and we can find some significant hits. But the bottom line is that although we can say we have done some things, we are just starting. And what I'm really sad about is, and I could not report this to you, for me, the one thing that I am the most passionate about, we have not done yet, which is activated kinases in these two subtypes, because there it gives you a straight shoe-in to molecular therapy of each subtype. There is no targeted therapy, so it's important for individualizing medicine, and this work can come out of India. It's happening right here. An ordinary day at NIMHANS, somebody comes, they have problems, people are still thinking what is happening there. You look at their MRI scan to try to figure out, and of course it's a series of steps, and they say, oh, there's an abscess. There's an abscess, people start to think about this, let's try to grow it. They quickly figure out it's not bacteria. So they do microscopy, they do culture, they also get CSF. They're looking for clues to figure out what it is. And here I'm going to talk about more infectious types of things. But in a special medium that is used, and this is the way it looks, there are these colonies that they look like yeast. Gram staining. So what I want you to remember is, just like diffuse and intestinal, the looks are not going to tell us what it is. And we will come back to this theme again. So it's basically a failed identification, and it was sent to a referral lab at PGI, and they take 3 to 4 weeks minimum to tell you what it is. So after 40 days, it was identified as cutaneous trichosporin, also known as trichosporin, by PGI 20 days later. It took some time to work up here, about 3 weeks and 3 weeks from them. So total time, 30 to 40 days, and we think we know what it is. But what we decided was, as a research study, the day the abscess came, we said, I want to prove that in one day, we can get the identification. So we started doing genomics, and we started doing proteomics. And in so this is just showing you how the sample was split. It's not going to be used for clinical reporting. In 24 hours, we nailed the problem. One single run on a high-end mass spectrometer, where we are sequencing the peptides, we could tell that this is cutaneous trichosporin oleaginosum. Okay? I won't bore you, but these are the kinds of data that come out. It's a mass spectrum of the peptide. We are sequencing hundreds of peptides. But if you eat meat, and if you're eating a burger, and you got to really say, is it goat, or pig, or beef, or some other animal? How will you figure this out? And I don't leave people in my lab until they can just recite in their sleep that really the only way we can identify it is we need the genomic sequence. And they always fail because they think, oh, we are doing proteomics in the lab, so the answer is proteomics. And I say, you fail. DNA, the genome sequence, will tell us what we have. So here, there was something that was lingering that was bothering us. Because we had these hits, but they didn't explain some of the data. So what we are also doing is, this is bedside nanopore single molecule sequencing. We have hundreds of thousands of reads, and we could take that, and this can be done very rapidly. People use it for Zika virus in Africa, Nature study. We can do it in Bangalore, right here. And now the story became different. So we were not sure, so we said, let's do Illumina sequencing, let's bombard it. After the bombarding, what we find is, we have 24 million reads from a culture. Now, this is not the sample, this is the culture. Only 6 out of 24 million reads are mapping under whatever parameters you choose. And then we say, okay, what we were thinking, cutaneous trichosporin, that species, it was not identical. It was similar, very, very similar, but not the same. So now we realize, okay, we're talking about one case, one clinical case. We have just discovered a new species. Same genus, different species. A lot more work is ahead, but what we have done now is these are our computational people. Again, new species, a new draft genome, 53 MB. Genome predicts 12,000 genes. You have to do that at that stage. But proteomics doesn't predict here. We are using that sequence, and we have done RNA-seq also. So we are now pushing forward for this. So what have we accomplished really? So this is the kind of a title that our paper would have, that we have done whole genome sequencing and proteomics to figure out we have a new species, all coming from one isolate. So you can see where we are going. India is the TB capital of the world many, many years ago. I don't know if you have heard of this place called Chengalpattu in Tamil Nadu. As a medical student, I was very proud. Chengalpattu trials. BCG was proven to be very useful in this condition. And yes, TB capital should do something. But I think in many, many intervening years, I think we lost the edge. We still are the TB capital, but I think this title is not really making me proud. But what can we do with what samples we have? And I'll just remind you what exactly happens. It happens every single day at every single hospital. People are assessed for the most common form, pulmonary tuberculosis. You have an X-ray, there's something suspicious, something that is cavitary, might be this. And you take the sputum, and you do what is called acid-fast staining. It's a messy process. Technicians in labs do it. So it is acid-fast. That means if it's resistant to acid in this decolorization step, then what you have is this. Acid-fast bacilli are there. It doesn't tell you it's Mycobacterium tuberculosis, what you want to know. There are many other related mycobacteria, atypical mycobacteria. So the real definitive identification is, and now that definitive is wrong, DNA will tell us, other things are almost getting there, but you have to do a shortcut which takes 10 to 15 days, and LJ slant which takes 4 to 6 weeks, 6 to 8 weeks. So you've got to wait for all that time, and then you're still not done because you need to do drug sensitivity. But people start presumptive treatment when they have, this I think I want to highlight. In India, this may not happen in many other places. People will start presumptive treatment which takes 6 to 9 months. 6 to 9 months of treatment the day they see this result. What also I won't tell you, but I just am going to tell you, you will not like it, is even if this is not seen on clinical suspicion, because clinical acumen is important, and I rebelled against this because I thought clinical acumen should be valued, but it's not everything. Sometimes you can be younger and you may have better ideas and you may be right. So if you don't grow anything, you can still treat, and that's the physician's prerogative. But 6 to 8 weeks. What we are saying is take the sputum, let's go to town as molecular detectives. Never done before experiment. In the sputum, you've got human cells, human protein. We are taking that, we are sending it through the high-end mass spectrometer, we are sequencing peptides, and a technician has to go through to look at acid-fast bacilli. We can say, oh, there are all these peptides that are coming that correspond to MTB peptides, and we can distinguish based on the peptide sequence whether it is typical Mycobacterium tuberculosis or many of these atypical mycobacteria. So we have identified Mycobacterium tuberculosis based on these proteins, which are specific for Mycobacterium tuberculosis not found in the others. What about genome sequencing? Well, we use those technologies, and negative samples were negative. These are negative by culture, so we are using them as our control. You saw it, but you didn't see it. So let's go back. There was a question mark. We have 1,000 reads that map to MTB in one sample that's labeled negative. Every single day this is happening. That is actually a TB case that was missed, and we have identified this. So again, we need to improve diagnostics. These are positive samples, and one of them was not positive by smear, but it grew out. So again, and we are taking the one that was negative by smear, actually is positive by whole genome sequencing. And again, like I told you, this is not rocket science. You know you can do this. But it's just application of that technology. Another day at NIMHANS, you are looking at these images after a lot of workup to try to say, is meningitis, maybe it's tubercular, maybe it's cryptococcal, maybe it's bacterial. Maybe it's something else. So how can we confirm the causative diagnosis? So we said we need to go direct from specimen diagnosis is not a common thing because of the human contamination in all these samples. I want you to understand before you'll realize why am I excited. These are new ways of doing things, not done before. So what are we seeing? These are MTB CSF from patients with MTB, but when we look at what we got based on our analysis, this is not tuberculosis. So we go and start reading now. I had never heard of this organism. Now we say, oh, 1991, there was a paper, Chimoidae. Then there's another paper now. Now it's becoming an emerging mycobacterial illness, which has a completely different drug resistance profile. So this patient actually doesn't have mycobacterial tuberculosis, has something else that is being talked about in the world. A lot of attention is being focused. How did we get to it? When we were trying to use it as a positive control to say, okay, we can find it. So we use proteomics and genomics to identify these. And I think this is important. People are talking about, there's so much talk about single cell sequencing and all that stuff. Clinical application. That's the power of mass spectrometry. DNA is even more powerful. 10 cells, mass spectrometry, we can identify. We know these are bacteria we have diluted as a synthetic experiment, 10 bacteria, and we can say, oh, many proteins, many peptides, what we have. We can do now, we have done the same with CSF for other organisms, because what we want to say is, can we do a differential diagnosis? Often you don't know what is the causative organism, but you are sure they have meningitis. And yes, there are some differences in clinical profiles. So here, again, we can say we are taking many of these samples. We have found Cryptococcus. Now we are saying let's do whole genome sequencing. Let's continue to do proteomics in that context. So now we start to do, these are our initial pilot experiments. Look at the one in red. These were Cryptococcus samples. They were culture positive, reported as Cryptococcus. It's Candida. We all know from the spelling, Candida is not Cryptococcus. It's very different. Treatment is different. This is what our being molecular detectives told us. And now you're saying, oh my gosh. What is happening? What I want to remind you is, I don't want to scare you. This is happening. It's true. I didn't make up any of the data. It's happening every single day. Every single day, in every single lab, at some level, some of this is happening. We are in the 21st century. We need to do things better. We already have these methods that many of you are using routinely in your kinds of experiments, changing the field. So, but as you walk out of the room, don't tell people about this. Because these are pilot studies, we are going to expand the scale, and we now need to say that these methods that I'm claiming as a research tool are giving us an output that we didn't have actually work when we do a test drive on hundreds to thousands of samples, and deploying in clinical practice is not that easy. And one of the reasons I moved to Mayo Clinic was that they have something called Mayo Clinic Labs, where they love to develop new diagnostic tests, and I'm a pathologist, and I want to do everything in my power to make this more objective. And these molecular tools are getting us there. And this is my last slide. Things I didn't get to tell you about. Thank you for listening and in this time, but I think this is a lot of the other exciting stuff that is evolving. We already have data on some of these, using CRISPR to make models for neurological disorders and cancer, to study genes where we don't understand them, but you can use CRISPR. And we have all the molecular tools to study signaling, including mass spectrometry, iPSC generation for, again, you can choose. And something that we are known for is developing new methods. And I heard some talks about protein-protein interactions. And this is a newer way to look at protein-protein interactions, is by proximity-dependent biotinylation. And finally, you hear a lot about circulating tumor DNA. It's all over the press, New York Times, Nature Science. What you hear less about is something that already is an FDA-approved test, which is circulating tumor cells. In some cancers, breast cancer is a major one, where the simplest thing that you can do with circulating tumor cells is count them. How many are there? And that is actually guiding treatment. But what we want to do is we want to do genomics of that. We want to understand, we can grow them, and we can do drug responses. And thank you for your attention.
Thank you, Akhilesh. Thank you, Akhilesh, for sharing very interesting data and also your experience setting things up. As we don't have time for questions, I'm sorry I have to stop here and invite Professor Satyajit Mayer for his talk. We're now all set to hear about the fascinating cell membrane.
There's a what do you call it, a series of open connections here, and a glass of water. Not a good idea. Don't feel like being fried for lunch. But before I begin, let me say that I owe a lot to the to the Wellcome Trust, first of all, for supporting me as an early career fellow. Wasn't an early career fellow, was a senior international investigator when I first came back to work in India. So my association with the Wellcome Trust and its incredibly generous and open-ended funding began in the early 2000s. Subsequently, the India Alliance came about, and since I was so grateful to the Trust for having put their faith in the research that we do, or we have done. I was involved in many of the efforts of the India Alliance in the beginning and subsequently. And have watched the India Alliance take off in an incredibly exciting way, and felt that after having contributed some of my time on many of the committees, I felt that I could also indulge myself in approaching them for a fellowship, which is perhaps the last fellowship of possible in the India Alliance's portfolio, the Margdarshi Fellowship. And to my surprise, as Akhilesh also indicated, I was given the fellowship after a great deal of discussion in the committee, and but I must say it has been quite amazing. I'm not going to speak about what I have achieved in my fellowship. I'm going to tell you something about the science that we do, but I would like to make one gesture, and that is, I'm sure that will be shared by all of our fellows. When I came to this meeting, Shahid he said, I'm going to ask you for a favor. And when the guy who runs the fellowship scheme says, I'm going to ask you for a favor, you don't say no. And so I said, what is it, Shahid? And Shahid, we have done what you asked us for. So on behalf of all the fellows and of India Bioscience, Lakshmi, would like to present you with some mangoes from Bangalore. It's an extraordinary effort. They're all organically grown in GKVK right next to NCBS. So it's an incredible thing to have Shahid as the India Alliance head, and also to look forward to the new innings of the India Alliance and what it can do with additional funding from the government. And I hope that continues. And I hope India Alliance continues beyond the 5 years in whatever avatar it may take. But it's a very important organization for supporting science in the country, perhaps one of the few that delivers money on time. Many of you will know what I'm talking about. Yeah. So anyway, so I hope this is not going to count in my time, but I'm just going to start my talk about what I'm here on this podium to give this afternoon. It's the skin, to tell you something about the skin of a cell. And what, I mean, the skin of a cell is essentially what I'm beginning to call the membrane of a living cell. And this has come about because we've been able to image the living cell membrane. And imaging the living cell membrane has revealed that it's a functional active membrane composite. And these are words, but I'm going to unpack them as we go along and tell you exactly what I mean by that, because it's something that we've come about by through an engagement with just watching what happens inside a living cell or at the surface of a living cell. So many of you would have seen this picture of a neutrophil taken from a movie by the late David Rogers at Vanderbilt University, now part of many textbooks. It's a neutrophil running after a bacterium. Incredibly important. If it didn't do this, none of us would be sitting here, given all the bugs that we heard about a little while ago from Akhilesh. So this neutrophil now has engulfed that bacterium, but it has really purposefully gone after that little creature. And the point I'm trying to make is that this neutrophil has gone after this little creature because the membrane of the cell, not the DNA, not anything else, the membrane of the cell is able to sense where this bacterium is. The membrane of the cell is actually looking at this bacterium, looking at its, what do you call it? Oops, where is that? Ah, yeah, here you go. There's a new toy that I got, which is now watching the bacterium and engaging with both physical and mechanical cues to go and trap this bacterium. Physical cues are coming from its environment, it's avoiding all the blood, and the chemical cues are coming from the bacterium. So how does the membrane actually sense this? And this is because the membrane senses this by actually engaging in transferring the information from the bacteria to the inside of the cell in real time and modifying the experience of the cell so that it can go and engage with this bacterium. So one needs to understand, what is this membrane? What is this membrane? How is this little 4 nanometer piece of fluid bilayer able to, in fact, extract all this information from the outside world? So if you just look at a cell in a dish, a very unhealthy looking cell, it's got all these spikes, but it still has a membrane. The membrane of the cell, for many years, has been thought of as a fluid bilayer, 4 nanometer thick, with 2 leaflets, millions of different molecules, thousands of different species of lipids and proteins, arranged presumably in a random manner, thermodynamically equilibrated. Because this is the impression of the membrane that we all have, and actually I had too when I started looking at the membrane of a living cell. Subsequently, from the 70s onwards, we've modified our view of the membrane, and this is a view of the membrane in the beginning of this century, 2000, a review from one of my colleagues, Michael Edidin, where he says the membrane is actually a patchwork quilt of different sorts of lipidic environments that the cell has actually created for itself, or the cell generates. These lipidic environments have been thought to have come about by and have been termed in common parlance membrane rafts, if you will. What I'd like to argue is that the cell is actually engineering this lipidic environment molecule by molecule. And the story I'm going to tell you about this afternoon is about how the cell is engineering this lipidic environment. It is not using standard equilibrium-based phase separation methods in this complex mixture, but it's actually literally putting molecules, well, I shouldn't say brick by brick, but molecule by molecule into place. So what we'd like to understand is the rules and principles behind how this local regulated organization works and how that can inform the cell of in fact, how it can go running after a bacterium in this very complicated external environment. And of course, those are the consequences of having such an enriched surface or skin, if you will, to engage with. So our journey into studying this membrane started many years ago when, of course, this was the model that we had in front of us, and we wanted to interrogate this model of a patchwork quilt of molecules in a living cell membrane. So to do that, we put in a few, I'll borrow a term from Akhilesh, a few detectives, and these were proteins which were anchored to lipid molecules, proteins that were anchored to, it's all over the place, this device. The proteins that were anchored to the membrane via glycolipids, and those glycolipids now have long saturated acyl chains on them, and these were GPI anchored proteins. I mean, they're present everywhere in the eukaryotic sphere of life. On every cell that is a eukaryote carries these GPI anchored proteins. So we put fluorescent tags on these GPI anchored proteins and began to observe the distribution of these GPI anchored proteins. If the membrane were a fluid bilayer where things were happily swimming around, well equilibrated, these molecules would have told us exactly that. They would have told us that they were randomly dispersed on the surface of these cells. But if you notice the scale bar here, it's 5 nanometers, and to ask whether these molecules were randomly dispersed on the surface of the cells, we needed to look at these molecules in living cells at that resolution. So to do this, we built some technology many years ago. This was a technology that wasn't available at that time to image molecules in the living state at 5 nanometer resolution. And actually ask whether another molecule was present next to it at 5 nanometer resolution. And in fact, we used a method that we pioneered to look at the interaction between fluorophores at 5 nanometer resolution. I'm not going to go into the details of that method, except to say that if you in this next view graph, if you look at the fluorescence distribution of a GPI anchored protein, here is a GPI anchored protein attached to GFP. Its distribution is uniformly boring. It doesn't look like this fantastic mosaic that I described to you. In fact, if those mosaic structures were present, the GPI anchored protein should have looked in this clumpy distribution that those mosaics gave us, but they looked very uniformly boring. But when we switched on our proximity imaging tool, we got this incredibly rich looking picture where you had all these different hues, which suggested, and I'm going to again tell you what these hues indicate. These hues, if you look at a little patch of membrane there, these hues tell us that GPI anchored proteins in these deep blue regions are present in these extremely clustered patches in the membrane. In regions where you have yellow hues, the GPI anchored proteins are sitting there monomeric and dispersed. So the presence of these proteins in these extremely dispersed states, in the presence of highly clustered states of GPI anchored proteins, was extraordinarily unusual. I mean, in a membrane, you should not be seeing this. I mean, here you have a lipid anchored molecule on the outer leaflet of the cell present in these tiny clusters in the membrane of a living cell. So this itself said that something unusual was going on. And if we could map the cell membrane in these blue and yellow or red pixels, where blue represents highly clustered, yellow or red represents unclustered, we should be able to get a sense of what the cell membrane is doing in space and time. And it turned out that this heterogeneity that we were observing was observed in our lab for the first time. And when we mapped this heterogeneity, this heterogeneity of distribution of clusters and monomers, they had very unusual modes. So as a physical chemist would describe this, these structures were incredibly improbable if you had a membrane if you had a fluid bilayer in which molecules were happily distributed in a random fashion. And they should not be happening. Not only were our methods revealing such heterogeneities, but subsequently many different techniques, especially the new super resolution techniques that became available to us in the last 10 or 15 years, have now revealed that these GPI anchored proteins are sitting clustered in the cell membrane. But I must say the experiments that we had done were in living membranes, and I think that provided us a very special glimpse of what was happening in the live cell. There are all sorts of properties of these clusters which now have revealed to us a very special characteristic, which I'm just going to now indicate in one movie here, where what you see in green are GFP GPI. When the cell makes these blebs, so every cell, if you bring them down onto a surface when they're in solution, first make these little blebs. And in these blebs, the GPI anchored proteins are present as very diffuse monomers. And experiments like this indicated to us that the clusters of these GPI anchored proteins were being maintained when the membrane was in fact in contact with the rest of the cell. And what is the rest of the cell that the membrane is in contact with? And that's the actin cytoskeleton. The cytoskeleton of the membrane of the cell is actually supporting this 4 nanometer fluid bilayer. And when the cell membrane engages with this 4 nanometer fluid bilayer, these clusters appear to form. But at that time when we were making these measurements and had encountered the possibility that the clusters of these GPI anchored proteins, remember they are outer leaflet lipid anchored molecules, engaging with the clusters of, engaging with the cytoskeleton at the inner leaflet, when they when the membrane was connecting to the cytoskeleton, what we knew is that diffusion of proteins in the membrane was affected by this engagement with the cytoskeleton. But the structure of molecules and their organization should, in principle, not be affected by the cytoskeletal armature that was present underneath the membrane. And a lot of this work was something that myself and a physicist colleague of mine, Madan Rao, were discussing, and we were looking at the analysis of all these distributions of these molecules in the membrane, when his student Kripa came up to us and said, look, you guys keep talking, but I'm going to tell you how these clusters might form. And what she found, and what she, being a physicist, she just said, well, let's being a theorist, I must say, not just a physicist, but not an experimentalist, but a theorist, she said, well, let's consider, for example, that you have in this complex actin cortex one more layer of complexity. That is, you have filaments of short actin and motor proteins that are engaging with each other. And if that happens, you will generate these tight clusters of actin being driven into these little aster-like configurations. And these aster-like configurations are essentially creating these clusters of molecules in the membrane. Of course, being a physicist and a theorist, you have the liberty to postulate anything you want. But as a biologist, we had to ask ourselves: is any of this possible? And what Kripa did was provided a incredibly coherent sort of theoretical explanation for all the experiments that we had done. The quantitative aspects of our experiments were completely explained by the theoretical predictions that she had done. In fact, what she produced was this little view graph for us, which actually convinced me that she was on the right track. What she's showing us here are clusters of these little asters being formed in space and time, and that's precisely what we were seeing in our experiments. So given that she had proposed this, what we thought at that time was an outlandish idea, we decided to look for these little filaments of actin in the membrane. And that's literally the model that Kripa put together. I mean, it doesn't require sophisticated cartoons to tell us that this is what we should be looking for. And when we went looking for these actin filaments, in fact, when I say we, it was Suvrajit Saha in my lab and Thomas Van Zanten. When they started looking for these molecules in my lab, they found them. They found them in great abundance, that you have these short filaments of actin sitting underneath the membrane, templating, if you will, structures in the membrane that could contribute to the patterning that we were seeing. Okay, so bear with me with all this, because this is going to end up in some serious consequences for the cell membrane, and I'm going to take you through some of the observations that have led to those, will lead us to those consequences. So essentially, what we decided at that time, based on all the theory and the experiments, that we are looking at a membrane which is being templated by the active mechanics of actomyosin. And this membrane templated by the active mechanics of actomyosin contributes to a composite, if you will, of a membrane sitting, being driven by the modes of actin activity happening underneath it. Right. So, but you must remember that the GPI anchored proteins that we were working with, these structures down here, they sit at the outer leaflet, and our actin is operating at the inner leaflet. So how does that connection being made? And there, we worked with a really amazing chemist, Ram Vishwakarma at the IIIM in Jammu in Kashmir, where he has labs. He built these lipids of different chain lengths, and which allowed us to make the following statement, that the GPI anchored proteins were connected to the actomyosin machinery of the inner leaflet through their interactions across the bilayer in a manner which creates little patches of membrane, of liquid ordered membrane at the sites where the actomyosin is engaging with the membrane. So at those sites, the GPI anchored proteins build these little liquid ordered domains in the membrane. Of course, and we did some all atom simulations of the structures of the lipids at the outer leaflet and the inner leaflet in the presence of cholesterol, and those indicated to us that these little nanodomains were being formed. So we looked for these nanodomains, and sure enough, here again, Suvrajit explored the membrane with new probes that we had built to look at the ordered regions of the membrane. And what he found was that the places where the GPI anchored proteins were forming these clusters, those patches in the membrane, in fact, were patches where ordered domains were being built. So this, again, convinced us that what we had discovered was a very general characteristic of the membrane, and that we could actually maybe generalize this into a new model for a membrane from these sorts of observations. So we put forward the idea that the cell membrane is indeed a membrane actin cortex composite, where one can think of three classes of proteins. One, of course, that corresponds to the diffuse, well-equilibrated Singer-Nicholasson type of membrane component, where the molecules are indeed well mixed. There's another class which is entrained by these actomyosin machineries, perhaps not just entrained by the actomyosin machineries, but it could also be entrained by activities that come from the outside of the cell. And that class of molecules were a separate class that we call the passive class. And there's a third class that we, based on these observations, and that energy was being consumed to create these structures, it didn't take us much time to postulate that there must be a third class. And I think this is the most important class, a third class of molecules that we call the active class, which indeed regulates this whole system. Again, this was driven by the idea that if these structures are being created because of the consumption of ATP, actomycin is, of course, a hugely ATP-intensive system, so if they must also be regulated. So we started looking for these regulators, and the first regulator we came across was the integrins. And now if you think back to the moving cell, moving cells require their integrins to be engaged, and these integrins are things that can drive, if you will, the changes in these membrane organizations. So integrins, in principle, have the following activities that they engage with in the inside of the cell. They engage with adapter molecules. They engage with the actin-binding proteins. They engage with kinases, GTPases, actin nucleators, and myosin regulators. All the necessary ingredients for creating these patterns in the membrane. So with that hypothesis, we went about looking for reasons to come to this conclusion. And what we did, when I say we, it's Joey. What he did was he put cells down which were in suspension onto surfaces that had integrin ligands, the fibronectin, for example, fibronectin. And sure enough, the cell membranes now suddenly begin to exhibit this blue aspect. So what he discovered was that there's a whole signaling pathway that you can see on this slide here that starts with the integrins at the top, and then enters the signaling cascade of integrins, leading to the generation of an actin filament and myosin motors. Oops. So now we find that this monitor seems to say 58 seconds to explode. No, I'm sorry. It says something very funny. But anyway, so the idea is that we've now established a completely detailed molecular machinery for signaling receptors to engage with this actomyosin templating mechanism beneath. That signaling machinery involves integrins of different flavors. The signaling, for example, fibronectin receptors activate such a signaling system. Other ligands of integrins, laminin, collagen, vitronectin, in fact, do the opposite. They inhibit such a signaling mechanism. But then Joey decided to do what one should never do, which is one more experiment. And what he did was he went and put the cells on a fluid bilayer, which had integrin ligands on it. And he found that now the cells activated their integrins, but did not make any of these changes in the membrane that we were looking at when the membrane was actually coming down on a glass surface that had been coated with fibronectin. Fortunately, he did these experiments in the lab of a collaborator in Singapore. And then when he came back with slides which had bilayers that had some defects in them, or some little chromium little pillars that we actually engineered as defects. And those pillars now trapped the integrin ligand, and the trapped integrin ligands now dramatically activated the membrane and its reorganization at the sites of activation. This indicated to us that the integrin system, when I say us, it was Ambika, she said, well, integrin activates a whole cascade of mechanochemical transducers. The integrins now activate because of their engagement and exerting some force on the surface, they activate these mechanochemical transducers like vinculin, and vinculin now generates a mechanical signal that gates the production of these specialized membrane domains. So I'm not going to take you through the details of this, except to now summarize for you that a upon activation from receptors or these active molecules like integrins, the cell membrane is modified in the vicinity of these receptors by both chemical and physical cues that are being sensed by proteins at those locations. Now, of course, this all sounds very well, and of course can get published, but then, why should the cell care about this at all? Right? I mean, I told you that the cell is acting as an adaptive sensorium. The cell membrane is acting as an adaptive sensorium, but how does this contribute to that sort of activity? So to ask that question, of course, we needed to perturb the way the cell is actually generating this sort of a patterning mechanism. And here again, we had some clues from the time course of the generation of these structures in the membrane. What we found was that the cells, in fact, produce these clusters just before they exhibited an explosive spreading activity. Okay, so and sort of some quantification to confirm that. And then we disturbed the production of those little clusters of GPI anchored proteins by using mutants that prevented the expansion of the chain lengths of those GPI anchored proteins. And we used cells that were no longer able to build these clusters in the membrane. And those cells now, if you look at them from a perspective of the spreading reaction, I guess I don't even have to show you what the mutants are, or tell you which ones are the mutants. So the ones on the left, of course, are the wild type cells. The ones on the right are cells that have been rescued, where the mutants have been rescued, and the ones in the middle are cells that just simply cannot spread. So putting all this together, we actually figured out how the cell uses some of these patterning machineries in order to execute some function. So the cell, in fact, upon engagement with their ligand, generates this reorganization of the membrane. That reorganization of the membrane, in turn, recruits a machinery which then activates the next step in the spreading of the cell, because the integrin receptor recruits a machinery that's dependent on the production of these domains for their spreading reaction. The spreading reaction is again an actin-based reaction, which is coupled to molecules that sense the change in the membrane order, and therefore this cascade of activities from integrin receptor signaling to the spreading of the cell or cell migration is directly coupled to the modification of the membrane of these living cells. This would not be that interesting if it were only the integrin receptors that were doing this. In fact, I've just got one more slide. Yeah. So the integrin receptors now act as, if you will, an exemplar of many signaling receptors that sculpt their membrane for such functional purposes. Many different signaling receptors activate the same sorts of machineries and indeed will modify the membrane in precisely the same ways. And they will engage in specific kinds of crosstalk using the membrane as an adaptive sort of surface. So here's one example. The signaling outcomes, in fact, of a GPCR are also modified by precisely the same sort of membrane environments that the integrin receptors can regulate. I'm again not going to show much more detail, but just now summarize for you what I think we've found is that the cell surface is a finely tuned actin membrane composite. It's a living, breathing skin that drapes the cell, and it consumes energy in a manner that creates sort of this adaptive sensorium. There are extraordinary properties of such an adaptive sensorium that we are exploring. I mean, they have implications for a number of different contexts. And in fact, I'd just like to leave you with one idea that what we are seeing is an exquisite orchestration of the regulation of membrane organization through these signal-dependent machineries, which in turn feed back onto the functions of these different molecules. There are a number of course, open questions, but I think this observation changes the way we look at that flimsy little 4 nanometer bilayer, and now consider it as a living, breathing entity that is able to modify itself in real time and modify the functions of molecules that are present within it. So I'm just going to leave you here with with an acknowledgement slide of all the different people who helped in this journey. But I would be remiss if I didn't point out some key people who've been part of this work: Joseph Matthew, Thomas Zanton, Chandrima, Subrajit, and a number of, and Ria, who were essentially the architects of the work that I've shown you this afternoon. Collaborations, without collaborations, none of this would have happened. Ram Vishwakarma, a brilliant chemist, and Madan Rao, an extraordinary soft matter physicist, have been part of this journey right from the beginning, and nothing would have happened without their engagement. And thank you all for your attention.
Thank you, Jitu, for the very nice story. In the interest of time, I suggest we don't take questions now, but I would request Jitu and also Akhilesh to be available for discussion over lunch. I now invite Shahid to give the closing remarks.
Okay, so we come to the end of another exciting meeting, and what I feel is that over the last 6 years that I've been with India Alliance, every meeting keeps getting better. That's good. We must keep improving. We purposely kept some exciting stuff for the last day, and you can tell by the audience that Akhilesh and Jitu can pull crowds. And in a sense, when I look back at the Margdarshi program, which is no longer operational, the 5 Margdarshi fellowships that we gave over a period of 7 years that the program was in operation, every single Margdarshi fellow is beginning to make a difference. I mean, you saw the 2 talks today. Coffee may not have woken you up as much as Akhilesh woke you up. And of course, people like Anura who work on nutrition, Vaskar who works on cancer. Vaskar is changing the way cancer treatments are done. Anura is changing food policy, nutrition policy in the country. Jitu, you've seen how deep the intellectual engagement was with cell biology, and we have one other Margdarshi fellow, Sandhya, of course. How can I forget Sandhya? Who is also not here, but Sandhya is building bridges using her Margdarshi fellowship. She is building bridges between basic science and clinicians by running a program between Indian Institute of Science and CMC Vellore. So, not to say that only our Margdarshi fellows are making a difference. I think we've seen through this meeting the exciting talks all the way from a research training fellowship recipient all the way to intermediate, early fellows, intermediate fellows, senior fellows, and Margdarshi fellows. So I feel that really the best part of the program are the fellows. And so I would like to thank all the fellows who are here, who are not here, for all the exciting work that you do that really makes us look good. And the committee members who select these fellows, because without their very critical insights, this would not be possible. And Akhilesh and Jitu both said that they were very surprised to get the fellowship. And I can now share that the committee at that time said, you know, we don't really understand what these guys will do, but I think they'll do well. You know, I think that has been proven that the critical insight and experience that committee members bring to the table is the result that we see in front of us. I would also like to thank all the speakers, whether they were fellows or invited speakers, for the very high quality of your talks. And you also helped us stand up and think. So I think that's very, very important. We can get very engaged and drill deep into what we are doing, but some of the speakers woke us up and said, look, you need to look at the real world as well. So thank you, everyone. I would also like to thank the staff at India Alliance. You work extremely hard to make events like this possible. You work behind the scenes. So I would like to thank everyone, and also let you know that tomorrow is not Monday is not a holiday. So could I have the India Alliance staff stand up, and everyone else, please give them a big hand. Our funders don't just give us the money. They also support us with processes. They support us with challenges. And I can tell you that there are many challenges. They also help us look at the future. And this is something you would have noticed when both Renu Swarup and Simon K. spoke on the first day. So India Alliance over the years has tried to transform itself from a funder to an influencer. And this is a trajectory that we really would like to be on. We would like to influence the ecosystem not by funding very high quality research, but also help the ecosystem in other ways. And you would have seen the research management initiative, IRMI, the new initiative that we have launched, which is the India Science Media Fellowships, bringing journalists together with scientists, international partnerships like the partnership with EMBO, and the new partnership that we are starting with Cancer Research UK. They're all terribly exciting. They also give us the opportunity to keep learning. And I think this is my key message to the fellows also, to keep learning. I mean, the world around you is changing very fast. Akhilesh showed what a nanopore sequencer can do at the bedside. Nanopore sequencers are now the size of a cell phone. They can be loaded on a drone, and you will see in a few years that they are going to be continuously surveying the environment through DNA sequencing. That world is going to unravel before you know. So keep learning, keep thinking, and continue to be the leaders that you already are. What does the future look like for us? Well, we have 5 years of funding. What after 5 years? Should we diversify? Should we continue along the path? And I think one of the things that we will look at very seriously in this 5-year period is to bring other kinds of funding into the program. especially private funding, philanthropic funding into the program. That will allow us to face new challenges, and it will allow us to learn new things. One other thought that has occurred to us, and this is also something that Renu said on the first day, can we use this platform to build something like a South Asia Alliance? We have a colleague from Nepal at this meeting. We will expand that engagement to neighboring countries and see if this is something possible in the future. And also, I can think about these things, and I can, put these into words, especially if I know that I'm not going to be around to implement these. These are in the long-term future. So it's always good to make a promise that you can't keep. And we see that happening all around us. So say, hey, why not? So anyway, we've had a long meeting. We had an intense meeting. So let me again say thank you to everyone for being here, for being part of this excitement. Continue to advise us, continue to engage with us, and do well wherever you are. Thank you again. Thank you very much.